really easy questions towards the end of the final, so if you get stuck on a hard one early, then you might not have time to do the really easy questions towards the end. Okay, so there will be 20 bonus questions, and they're sprinkled throughout the exam, so it's not like all the bonus questions are at the end, they're just sprinkled throughout. Try to bring a couple number 2 pencils, and you guys, it really does help to get sleep the night before, try to eat something before you come in, right, because it's a marathon exam. So you're not sprinting on this one. Okay, let's see here. And then folks, if you want your final exam data link sheets sent to you, or if you want your unknown reports sent to you, I usually don't have them done by the time of the final, please bring a stamped self-addressed envelope to the final, and I'll stick in your data link sheets and your unknown report. Or if you want, you can just come by my office at the start of spring semester 2020 and pick them up then. Okay? All right, you guys, so we are on this wild and crazy journal through the adaptive acquired specific immune system. And folks, you know, as I was writing the final, I was like, oh, I have to make sure I tell them about passive and active immunity. And so you guys, down at the bottom of your little host defenses concept map here, you guys, under adaptive immunity, there's two different ways to classify it. One is passive immunity. For example, the classic example is if you receive antibodies that you did not make, right? So 2 natural examples of passive immunity would be mom transferring her antibodies across the placenta to her baby. So that transplacental transfer of what we call maternal antibodies is a natural example of passive immunity. And a second example, you guys, that's really cool is that when mom is breastfeeding, any mammal that's nursing their young, mom mammal is transferring antibodies into her breast milk. And that's another example of passive immunity, maternal antibodies. An example, you guys, of artificial passive immunity would be if we inoculate a patient with antibodies, say against a pathogen or against a toxin. Like, for example, if you had a patient suffering with botulism or tetanus, you could inject them with antibodies against the toxins, right? So we call that antitoxin. So that's another example of passive immunity. And then, folks, what we're talking about for the rest of the lecture is all active immunity. This is when you're going to make your own antibodies, or you're going to make your own cells for cell-mediated immunity. You're doing it. You're actively involved in the immune response. Okay, so that was just a little tidbit there. And then, folks, you will recall that we are opening up this lecture on adaptive specific immunity, saying that if you don't learn anything else, we want to understand the key role of T helper lymphocytes, also known as CD4 positive T helpers. We want to understand their key role in coordinating all the responses of adaptive acquired immunity. And we said that the way the T helpers are going to coordinate all the other cells of the immune system is by producing chemical messengers that we're just going to generically call T helper cytokines. Okay, and so you guys, just up on the board, I'll try to squish it into this little space. Again, we had started on this, and we had laid our paper landscape version. And our key player, the conductor of the immune system here, you guys, is our CD4 positive T helper lymphocytes. And we had divided our acquired adaptive immunity into 2 major branches: humoral immunity, protection provided by antibodies. And we had said that antibodies primarily protect us against extracellular pathogens and extracellular toxins. And the reason that antibodies aren't good, we're going to say, for intracellular pathogens or intracellular toxins is antibodies can't cross the cell membrane to get at the pathogen or the toxin. Okay, and so folks, just really quickly, if humoral immunity is provided, the protection is provided by antibodies or immunoglobulins. Did we talk about who makes the antibodies? The B lymphocytes, right? And we said for the B lymphocytes to respond in the most efficient manner, what chemical messenger do they need? Yeah, the T helper cytokines. So we were just cartooning, you guys. T helper cytokines is this blue arrow. So these are the chemical messengers the T helper makes to help the B cell carry out its function most efficiently. And folks, you'll recall that the T helper cytokines are essential for that process called class switching. And this will make a little bit more sense by the end of the lecture. And we said in class switching, we want the B lymphocyte to switch from making the first antibody, IgM. We want it to switch to making the best antibody that has the greatest variety of functions. And which one was that? IgG, good. And folks, IgG, we'll discover, is the only antibody that can cross the placenta. So it's the only antibody that mom can transfer across the placenta to the baby, and that's really important, right? And then furthermore, you guys, we were saying with adaptive acquired immunity, a big advantage of it is the phenomenon called immunological memory. So what do we want our B lymphocytes to make so that we'll have that immunological memory? Good, so we want to make B memory cells. And folks, can the B lymphocytes undergo class switching? Can the B lymphocytes make B memory cells if there aren't any T helper cytokines around? No. Yeah, so this is crucial. Okay, so this requires T helper cytokines. So that was the humoral arm. And then, folks, what we're going to do, just again, very, very superficially, is we're going to look at the second arm of adaptive acquired immunity, CMI. And what does CMI stand for? Good, cell-mediated immunity. It's not involving antibodies. It's going to involve like killer cells. And this is the arm of the immune system that's going to deal with intracellular pathogens, intracellular parasites, intracellular pathogens. And you guys, there's going to be two cells that we'll be describing. And so the first cell is another T lymphocyte, but these have a little bit different function. They're called CD8 positive cytotoxic lymphocytes. And what we're going to see is that their job is to patrol our bodies, looking for our cells that have been infected with intracellular pathogens. And a classic example, you guys, would be a virus-infected cell, right? So their job, excuse me, their job is to identify our cells that have been infected, say, with viruses, and then they destroy our cells. And that might make you feel really weird. It's like, what's the immune system? What's the immune system doing harming our cells? You guys, why would that make sense? Why would you want to kill one of your cells that has a virus replicating in it? So it can't replicate, right? Yeah, and if you can kill the infected cells early enough, then you'll stop the virus from replicating. It won't spread to other cells, right? So, but to do that, to carry out that function, you guys, what kind of chemical messenger must the T cytotoxics receive? T helper cytokines. So you guys, you can see on this exam, any question that deals with acquired adaptive immunity, if I'm asking you what's required for this process to occur, what's a good default answer? T helper cytokines or T helper cells, right? Good. And so again, folks, just and really superficial here, the T helper cytokines are required so the T cells can kill infected host cells. So, for example, we use a viral infection. So the T helper cytokines are required for the T cytotoxic cell to kill the infected host cells. But again, you guys, we also want that immunological memory. So what else will the T helper cytokines help the T cell do? Yeah, exactly. Right? So we want to make memory cells. And again, folks, the T cytotoxic cells can't do any of this unless there's T helper cytokines present. Right? And then the last example, you guys, of how crucial the T helpers are, we're still in cell-mediated immunity. We're going to use the example of a resting macrophage. And this poor resting macrophage has been infected with one of those facultative intracellular pathogens or parasites. And the classic one we use, you guys, is Mycobacterium tuberculosis. So this macrophage has been infected or parasitized by, for example, Mycobacterium tuberculosis. So we would say the macrophage has been infected or parasitized when they cannot kill the pathogen they've ingested, okay? So these resting macrophages, they have really low phagocytic ability, but in a weird sort of way, unfortunately, they can phagocytize. So, for example, if this was a macrophage, say, in our lungs, and we inhaled Mycobacterium tuberculosis, the macrophage could still ingest them, but it can't destroy them because it's resting. It has to be activated. And I always think of you guys as myself in the morning when I first get up, and I'm just barely awake until I have my first cup of stiff coffee, right? Like high octane, high caffeine coffee. And then I can think, right? Well, I always think that this poor resting macrophage needs the equivalent of a stiff cup of coffee. And who do you think is going to deliver that stiff cup of coffee? Yeah, the T helpers. And again, folks, we think of those T helper cytokines. These are going to be the chemical messengers that will wake up the resting macrophage and turn it into what's, I love this term, you guys, turn them into angry killers, okay? And so the T helper cytokines will turn macrophage into what are called angry killers. And what we mean by an angry killer is a phagocytic cell that is very efficient, very rapid at phagocytizing invading pathogens. And here's key, you guys. They're going to increase production of those hydrolytic lysosomal enzymes, right? Remember we talked about the oxygen-independent killing, right, of the hydrolytic lysosomal enzymes that will try to digest the pathogen. And then furthermore, you guys will see a big increase in the production of those reactive oxygen intermediates, such as superoxide anion, hydrogen peroxide. Remember, that was the respiratory or oxidative burst, the oxygen-dependent killing. So the T helper cytokines, and one, folks, you don't have to memorize this, but I'm just going to throw this in there. One of them is gamma interferon. And you might recall in non-specific defenses, we had talked about alpha and beta interferon and how they can trigger our cells to make those antiviral proteins, right? But we had mentioned there's a third interferon, right? So T helpers, one of the chemical messengers they can make is this gamma interferon. And I think of gamma interferon as high caffeine coffee for the macrophages, right? Okay. And so the idea is, you guys, by increasing those hydrolytic enzymes, by increasing production of reactive oxygen intermediates, now the activated macrophage can kill those facultative intracellular parasites, right? But again, you guys, this has to happen early, right? We have to have like a pretty quick response. Okay, so you guys, that's the big overview of the key role of our T helper lymphocytes. And you'll remember one of my goals here was I wanted you to understand why is it that if I get infected with HIV, right, it's going to replicate in my T helper lymphocytes, and eventually it might kill so many of my T helper lymphocytes, my immune system collapses, right? So I'll just give you some figures here, you guys. So if we're talking T helper lymphocytes, kind of the average in a normal immunocompetent human, strong immune system, it's about 1,200 T helpers per microliter of blood. But the problem is, you guys, is that HIV, if I get infected, HIV is going to replicate in those T helpers and is going to kill them. And so it might take a while, but it might be that in a few years, so many of my T helpers are destroyed. If I dip below, if I'm at or below 200 T helpers per microliter of blood, this is immune collapse. Now, why do we say immune collapse, or why do we say that HIV has crippled my immune system? So just think, you guys, if I don't have enough T helpers, can I get my B lymphocytes to undergo class switching from IgM to IgG? No, and we'll see, you guys, that's really bad, right? And furthermore, you guys, can I make any memory B cells? No, right? And then over on CMI, you guys, could my cytotoxic T lymphocytes efficiently kill my cells that are infected with viruses? Nope. And without my T helpers, can I activate my macrophages so that they can kill these facultative intracellular parasites like Mycobacterium tuberculosis, Listeria, Salmonella, Shigella? No. So what happens, you guys, is if I'm HIV positive and my T helpers are below 200 per microliter blood, now officially I have AIDS. And what does AIDS mean, folks? Acquired immunodeficiency syndrome, right? And again, folks, all the cells that are involved in adaptive immunity, they don't work anymore. They've lost their conductor. You know, often people talk about T helpers as like the conductor of the immune system. So the cells can't become activated. They can't carry out their processes normally. They can't make memory cells, right? So this is the heartbreak of AIDS, right? So, okay, so again, if we understand that key role of T helpers, and if we can understand why it is that HIV can cripple the immune system, then I think you've got like the most important part of acquired adaptive immunity, right? Okay, all right, guys. So now what we'll do is we'll go back to like kind of like the vocabulary slides, kind of like the foundation here. Okay, folks. So one thing we want to understand is that for our adaptive acquired immune responses to be activated, they have to have a signal. So any substance that will trigger adaptive acquired immune responses, and usually we use, as an example, antibody production, such a substance is called an antigen. An antigen. So antigens, folks, should not be self, meaning antigens should be something foreign, you know, that invades us from outside of our body, such as a virus or a bacterial pathogen, right? But you guys, is it possible, are there some diseases in which our own cells or our own substances that our cells make that can also act as antigens? Are there such diseases? Yeah? Where your own cells, your own substances trigger an adaptive immune response? Yeah, so what would we call that group of diseases? Autoimmune, self-immune, right? And those are diseases, you guys, that's going to cause a lot of harm to our cells, right? And it is fascinating, you guys, they think they're seeing an actual increase in autoimmune diseases. And if we have time, we'll come back and try to explore that a little bit. Good. So an antigen hopefully is going to be foreign, non-self, right? An invading pathogen, as an example, or maybe an exotoxin. Good. And then, folks, if we're talking about microbes as antigens, it turns out that a single microbe can actually make multiple substances that will trigger an immune response. So let's take a look at the next slide. Okay. So remember, folks, I said that our example of an immune response would be antibody production. And we're going to discover that the function of the antibodies is to bind to the antigen, the substance that triggered its production by our B lymphocytes, right? So, folks, if this cartoon represents an invading microbe, we'll just pretend it's a bacterium. What we see here, folks, is this antigenic bacterial pathogen. It has many different substances which can trigger production of different antibodies. And here, folks, the idea is kind of like the lock and key idea that we first introduced when we were talking about enzymes and substrate. Okay, so the antibodies are going to bind to complementary antigens that have a similar size, shape, charge distribution as is what's called the antigen binding site, which we'll get into here in a little bit. But folks, if this is a bacterial pathogen, what we notice here is it has here 3 different substances that are triggering unique antibody production. So the substances that the antibodies actually bind to, those are called antigenic determinants or epitopes, because you'll see that a lot in your readings. And folks, I'm so bad. I don't use antigenic determinants or epitopes. I just say the antibody binds to the antigen, right? But again, we just want to make sure that we recognize that a virus or a bacterial pathogen could trigger production of different antibodies to different substances of the bacterium and the virus. So folks, what we'll first then explore is humoral immunity. And again, folks, humoral immunity, oops, it used to be here, refers to fluids. And what's protective in the fluids? It's antibodies, also known as immunoglobulins. Okay. And again, folks, we said that for us, we're kind of oversimplifying it a little bit, but we're going to say that humoral immunity, antibodies and immunoglobulins provide protection against extracellular pathogens and extracellular toxins. So folks, as always, we want to look at the structure of our antibodies, immunoglobulins to understand function. And there's a lot of little detail to this, folks, so I hope I won't overwhelm you. So what we're going to use as our sample, our example antibody, is the antibody known as IgG. We're going to see there's 5 classes of antibodies that humans make. The predominant antibody in normal humans is IgG, and this is awesome, you guys, because we'll discover IgG is truly the best antibody. It carries out all the diversity of functions that we'll be exploring, and furthermore, folks, it's the only antibody that can cross the placenta from mom to baby. Okay, so we're going to use IgG as our kind of generic antibody, and one thing we want to know, folks, is that IgG, it consists of 4 polypeptide chains. 2 of them are long, so here's the long ones, and 2 of them are short. So the long chains are called the heavy chains, and the short chains are called the light chains. And I just think of it as like, okay, the heavy ones have more amino acids, so they weigh more. The short ones have fewer amino acids, so they don't weigh as much. So that's how I remember heavy and light chains. And then, folks, you'll see that because there's 4 different polypeptide chains, they have to have this very specific arrangement. So this is a great example of quaternary structure of proteins, where you have a protein with 2 or more polypeptides. The polypeptides have to be in a very specific quaternary structure for the antibody to function. Now, we can see we have this Y shape, and you guys, I always use like my body is an antibody. So we can see the antibody has 2 arms, and we can see that it has what's called a stem, right? And what's really cool, you guys, is at the end of the 2 antibody arms are antigen binding sites. So it's almost like our hands, right? So if you guys, if I don't offend you, can you be my invading pathogens? Thank you so much. And I'm an antibody, right? And I can bind to the surface of the invading pathogens, right? By my antigen binding site. So my hands are like the antigen binding sites of the antibody. Now, in a lab, folks, this would be a lab experiment. We can take a specific protease, an enzyme that can cleave proteins, and we could cut the antibody right here at what's called the hinge, and that would release 3 fragments. And 2 of the fragments, you guys, would be these antibody arms, and those 2 arms are referred to, let me write it up on the board. The 2 arms are referred to as the antigen binding fragments, and of course we use initials. So, okay, so here's my model IgG. Oh, and folks, I'm sorry, I forgot to tell you that the quaternary structure is stabilized by R-group interactions, and usually folks just put disulfide bridges up there for the R-group interactions, but again, R-group water is going to stabilize the quaternary shape. Okay, so guys, so if I cut my antibody right at the hinge here, it's going to release the 2 arms, and the 2 arms are known as the FAB fragments, so FAB, and AB stands for antigen binding. And that's because you guys will recall, we said at the ends of the arms, it's like the hands or the antigen binding sites, we'll have a groove of a specific size, shape, and charge distribution that can bind to complementary antigens or antigenic determinants or epitopes. So the FAB fragments, you guys, are the antigen binding fragments, and the antigen binding site is right at the tips. And we'll see on our antibody that it has 2 antigen binding sites, and we'll see that that's going to be important in function. Now, this bottom part, you guys, officially it's called the FC fragment, which is kind of, the official reason for C is it's called crystallizable, which to me is not very helpful. I want it to tell me about function, right? So I think of the C, you guys, is complement binding, for indeed, once our antibody binds to an antigen, then complement can bind. So you guys, let's use a little C for complement. So once the antibody binds to an antigen, now complement can bind to the stem, and the complement then will be activated. And you might remember, you guys, activated complement benefits production of inflammatory mediators, chemotactic factors to guide the white blood cells to the scene of invasion. We're going to make opsonins, right? And we're also going to form this really cool MAC attack, the membrane attack complex, right? So complement's going to bind to the FC stem. And then furthermore, you guys, it's on the FC that the FC region can bind to FC receptors on phagocytes. And we'll cartoon this. So if we can coat an invading pathogen with antibodies, right? So this FC stem, the end's going to be sticking out. And this is awesome, you guys, because our macrophages, let's say this is our macrophage. Our macrophages have what are called FC receptors on their surface. So these big red things, you guys, these are the macrophage FC receptors. So again, folks, this might sound familiar. This is another example of the process we call opsonization. When the invading pathogen arrives, if we can coat the invading pathogen with IgG, right, the macrophages now can really quickly, efficiently phagocytize, ingest this opsonized pathogen because it has receptors for that FC stem. Okay, and I think we have a cartoon here where we'll be exploring that a little bit more. So you guys do remember then that if I ask you about what are opsonins, we want to remember complement can act as an opsonin, and who else can act as an opsonin? Antibodies can, right? Coating the invading pathogen, and then the phagocytes can bind to those opsonins and really increase the rate of phagocytic killing. Okay, let me see here. Is that everything? I think so. Okay. So folks, just with it, kind of like that brief introduction then, we're going to look at some of the functions of antibodies, okay? And the functions, you guys, include the ability of antibodies to cross-link cells or cross-link pathogens, causing them to clump together. And that process, you guys, is called agglutination. And again, folks, it's related to structure. Because remember, each of the antibodies has two antigen binding sites. So each antibody can bind to two different pathogens and cross-link them, and then another antibody can bind and cross-link two more. So it forms these big clumps. You might say, well, how is that helpful? Well, this is awesome, you guys, because it prevents the pathogens from spreading. You make these big clumps, right? It literally weighs more, right? So it's going to prevent the spread of the pathogens. And furthermore, folks, this will also increase phagocytic killing, because now a phagocyte can ingest a clump of pathogens, not just one at a time, right? So that's awesome. Another cool process or function of antibodies, folks, is one you all know already called neutralization. What do we mean by neutralization? Blocking attachment, right? So you guys will recall we've talked about how antibodies can bind to the adhesins on the surface of a bacterium or on the surface of a virus, and thus block attachment. The pathogen can't attach to our cells, therefore it can't colonize us. And also, folks, do remember that antibodies can bind to the B subunit, the binding subunit of our exotoxins, and do the same thing, prevent the exotoxin from binding to the surface of our cells. And if the exotoxin can't bind our cells, it can't get into our cells. It will prevent harm, which is really cool. And then, folks, down here, the third little cartoon here, this is a nicer picture, folks, of how antibodies can act as opsonins. So just to decipher, this is our invading pathogen, maybe a bacterial pathogen. Here's our antibody, and notice, you guys, the antibody is bound by the antigen binding sites, and that's cool because it leaves that FC stem pointing outward, and that's perfect because now our phagocytes can come along, And what's this little turquoise box, you guys? That's on the phagocyte surface. What is that? That's the FC receptor, right? So now the phagocytes can use their FC receptors to bind to the antibody opsonized pathogen, and that really increases the rate of phagocytosis. It really increases phagocytic killing. Yeah, so this is awesome, you guys, because again, if we just really quickly review what else can act as an opsin. What else can act as an opsin, folks? Complement, right? So let's say we've activated the complement cascade. So we've opsonized our pathogen with complement, and in addition, we have opsonizing antibodies. Yeah. So we'll just make this up, you guys. This might be, say, complement, and that might be an antibody. So you'll notice that our phagocytes have receptors for both the complement and the antibodies. So in coding with both complement and antibodies, man, are we going to increase phagocytic killing, right? That's going to be an amazing response. Okay, so remember, folks, opsonization, preparing to eat. We're coating our invading pathogens in sticky substances to which the phagocytes can bind and then kill the pathogens. This last kind of concept map, you guys, the reason I stuck it in here was so we'd have a cartoon of how when antibodies bind to their antigen, say they bind to the invading pathogen, once they bind, complement can then bind to that FC stem, and the complement system will get activated, right? So inflammatory mediators, chemotactic factors, more opsonins, and what else? They could outright kill a cellular pathogen. Membrane attack complex, right? So the complement forms those cool pores and lipid bilayers. So it could be the cell membrane of a cellular microbe causing lysis. It could be the envelope of a virus. And remember, you guys, if we destroy the envelope or we harm the envelope, then the viruses can't attach to our cells anymore. It could be the outer membrane of a gram-negative bacterium. We can have membrane attack complex there. And then the complement can get in to the periplasmic space and then form membrane attack complexes in the cell membrane of the gram-negative. So again, you could just get outright lysis. Yeah? Okay, so those are the functions of antibodies, folks. And we wanted to go through the functions before we talk about the 5 classes of antibodies, because it turns out only one antibody can do all those things. So we just really briefly, you guys, very superficially, we want to talk about the classes of antibodies. Okie dokie. So the first antibody, folks, that our B lymphocytes make is going to be this really cool antibody called IgM, and it can be a single monomer, but usually in blood it's a pentamer, and pentamer is 5, so 5 parts, and they're so cool. To me, they look like little snowflakes. So here's our beautiful IgM pentamer. Again, the first antibodies that our B lymphocytes make. And oh my gosh, you guys, IgM is awesome at agglutination because it can actually bind to 10 different pathogens, right? It causes massive clumping, massive agglutination. It can activate complement. But you guys, there's some disadvantages to it. And one of the disadvantages is it's so big. It usually is found primarily within our blood. We don't find it in high concentrations in the tissues outside of the blood vessels where often the pathogens are. Another disadvantage, you guys, is it's not opsonic. And the reason is it kind of makes sense, right? If you're a phagocyte, right, and you're trying to, you want to bind to an FC receptor, see how all these FC ends are sticking inward, so they're not accessible for the macrophages, the phagocytes to bind to. So a big disadvantage is IgM is not opsonic, right? And another big disadvantage, you guys, is IgM can't cross the placenta, so babies can't get any of mom's IgM, right? So I mean, it's an okay antibody. I don't want to show it any disrespect, but it's not the best antibody. Now, who's going to be our best antibody? Yeah, IgG. And thank heavens, you guys, that's the predominant antibody. I think it represents about 70 to 80% of our normal circulating antibodies. So we would say the best one with greatest number of functions is going to be good old IgG. And again, folks, if we talk about diversity of functions, can you help me out, you guys? What are some of the functions of antibodies? Let's list them again. Agglutination. Okay, what else? Neutralization, awesome. Block attachment, good. What else? Awesome, you guys. Opsonic, it's a wonderful opsonic antibody. What else? How about what can bind to the Fc stem and get activated? Complement, yeah? So it activates complement. That's a huge advantage. And folks, I'll put a star by it because IgG is the only antibody that will undergo transplacental transfer. What is that? What a mouthful. Transplacental transfer, what does that mean? Yeah, mom to baby. Yeah, moms. Maternal antibodies transfer to the baby across the placenta. And that's so important, you guys, because the baby's immune system, even at term, the baby's immune system is really not that functional. And just imagine, you guys, if you're this little baby in this relatively microbe-free environment of the uterus, and then you experience a vaginal delivery. And just imagine all the microbes a poor little baby is going to be showered with. And just think, you guys, during the vaginal delivery, probably the baby is getting also exposed to mom's fecal bacteria, right? Not to mention all the bacteria on our skin, our hands, on every surface around, right? And if that baby can't make its own antibodies, that would be really horrible. But what mom has done ahead of time is transferred her protective multifunctional IgG across the placenta. So the baby already has mom's protective IgG circulating in the blood, the lymph, bathing all of the cells in the extravascular tissue. So what a beautiful, beautiful system, right? But again, you guys, only IgG gets transferred. And again, isn't that awesome? Because it's the antibody that can do everything, right? It can do everything. Good. And then, folks, another really cool antibody is IgA. And the one I want to focus on, you guys, is called secretory IgA. And secretory IgA, it's a dimer. It's made up of two monomers joined by this J chain is what joins them. And then it's got this really cool component you guys call the secretory component. So let's talk a little bit about secretory IgA, because it's awesome. Okay, so S is for secretory IgA. And we think of secretory IgA, you guys, as being really crucial in mucosal, can I spell mucosal? Probably not. Mucosal immunity. Because it's secreted by the epithelial cells of the mucous membrane. And so it's going to be really rich in that mucus blanket that coats all of our mucous membranes, the conjunctiva, the mucous membrane of the respiratory tract, the mucous membrane of the gastrointestinal tract, the urinary tract, the genital tract, right? And what's so awesome about it, you guys, is that it's a wonderful neutralizing antibody. And this is fantastic because if you think of our mucous membranes, you guys, really microbes from the outside have access to most of our mucous membranes, right? And so if we have the secretory IgA in that mucus blanket, the secretory IgA can bind to the invading pathogens. And then what's so awesome, you guys, that little secretory component there, it will bind to components of the mucus blanket. So it gets stuck in the mucus blanket, and then there's all different ways, you guys, we've evolved to get rid of the mucus blanket. We can cough it out, sneeze it out, we can cry it out, we could poop it out, we could urinate it out, right? And so with the pathogens coated with the secretory IgA, they get stuck in the mucus blanket, and then they'll somehow physically get removed, right? And again, you guys, so microbe can't bind, can't attach to our cells, no colonization. And so furthermore, you guys, not only is it fantastic in mucus secretion, it's also wonderful. It's present in colostrum and breast milk. So colostrum, you guys, is the first milk a mammal makes after giving birth. And it's oh my God, it's fantastic. It's so rich and thick. It's chock full of antibodies. Because the whole idea is the first meal the baby takes, we want to just feed as many of mom's protective maternal antibodies in that colostrum, that first milk, to coat the baby's intestine to provide passive protection, right? And you guys, it's not only that secondary IgA is going to be in the colostrum, it will also be in breast milk, right? After the first milk, after the colostrum is made, and then mom starts pumping out just regular breast milk, it's going to be really rich in secretory IgA, as is the colostrum. And this makes sense, you guys, because how do babies explore their world? How do babies explore their world? They put everything in their mouths, right? You know, you put a little baby down on the ground, they're putting, you know, handfuls of dirt into their mouth. So you can imagine, oh my gosh, you know, they're going to be at such high risk of intestinal infections, right? So this is so cool, you guys. So as long as the baby is breastfeeding, you can imagine like the surface of the intestinal tract is just coated in mom's protective secretory IgA, right? Isn't that awesome? There'll be other antibodies in the milk, it's like IgG in the milk, but you know, I always think of that secretory IgA as helping provide protection to the intestinal tract of the baby. Yeah.
So babies that are formula fed, do they ever gain those antibodies on their own, or is there?
So that's such a good question, you guys, because that's one of the disadvantages of a formula-fed baby. Is that now, and you guys, maybe there's been some advances that I don't know about, but I know that when our kids were little, as far as I know, the formula, because one of our babies, we were formula-fed the baby. As far as I know, a huge disadvantage is they aren't getting those maternal antibodies. And again, you guys, it'd be awesome if there was some technology where formula could have maternal antibodies in it. That'd be awesome because it's a huge advantage to the babies. It really helps protect against diarrheal disease. And kind of on that topic, guys, and I'm sorry, you know I go off on my soapbox thing, but I do have to share this with you, just so you guys are aware and you can educate. So the Nestle's Corporation, I think of them as chocolate milk, right? But they make baby formula. And there was a scandal that, and this was several years ago, that the Nestle's Corporation went into areas of the world and offered initially free formula to nursing moms. And the problem is once you stop nursing, your milk dries up, right? So my understanding is they gave free formula to the nursing moms, so the mom, they were convinced, oh, this is easier, which is not true. But anyway, and so the mom switched to formula, and then, you know, after the free samples are gone, formula, have you guys priced formula? It will bankrupt you. It is so expensive. So this was so unethical. I mean, there's always situations where babies will have to be formula fed, right? But if mom has a chance to breastfeed, that is really the healthiest alternative for the baby. It provides this incredible protection against diarrheal pathogens, right? And furthermore, folks, where this company was carrying out, I just don't want to have any Nestle's products anymore. So in a lot of those areas, the water was fecal contaminated, right? A lot of regions of the world, water is not safe to drink. So what do you need for formula? Water. So it was like that was absolutely unethical, because then not only is the formula expensive, but then the water that's being used to mix the formula has fecal pathogens in it, and the baby's not getting those protective maternal antibodies in the breast milk. So you guys, I'm sorry, stuff like that, I'm like, you know, you guys, why don't you do something that helps people instead of just trying to make all that money? Anyway, sorry you guys, anyway. So folks, if folks can, it really is best if you can breastfeed, and sometimes moms need help and support because it can get hard getting it started. But I think there are organizations, you guys, like La Leche League, is that right? That will provide help and support. In the old days, when every generation would breastfeed, you know, grandma or great-grandma could help a new mom learn how to breastfeed and kind of get over the tough parts. But yeah, in the 50s, there was a big push against breastfeeding. So some of the generations, the moms kind of lost the knowledge of how to, you know, get through those rough spots in breastfeeding. But you guys, seriously, it's like it is if mom is healthy and if the baby's healthy, breastfeeding is just amazing. It gives them a lot of advantages. So there's my plug against Nestle's and my plug for breastfeeding. Okay, so you guys, so let's move on from wonderful secretory IgA, then let's move on to IgE. And folks, IgE, it probably initially evolved as an antibody to protect us from large parasites like worms, okay? And this makes sense, you guys, because let's say you've got a big worm invading you. So we're going to talk about IgE. So IgE, so this is like helminth parasites, large parasite protection. And it makes sense, you guys, because you have a great big multicellular worm invading you, right? And you've got this little phagocyte. Oops, maybe smaller than that, right? So this is a little phagocyte. And this is the worm.
We're talking about IgE, right?
Oh, sugar, thank you guys. Oh, you guys, I cannot talk and chew gum. So thank you, I made a mistake on the board, folks, sorry. IgE, thank you so much. Okay, so folks, if this is like our little phagocyte and this is this great big parasitic worm, is that little phagocyte going to be able to ingest the worm? No, so phagocytosis isn't going to work, right? So this is where IgE is so cool. So what will happen is the IgE will bind to the surface of the worm. So this is our IgE. And then, folks, and you don't need to know these details, but I just think it's the coolest thing. We've got another type of white blood cell called an eosinophil. So I'll make great big eosinophils here, guys. The eosinophils can bind to the IgE because they too have Fc receptors for IgE. And then when they bind, they release all of these anti-worm compounds. So these are like proteins, which a lot like the membrane attack complex, you guys, we see these strategies repeated over and over again. So there's like proteins, they form pores in the worm and can kill the worm, right? Isn't that cool? So again, this is a really neat strategy for large parasites that are too big for phagocytic cells to ingest. You coat them with IgE, and then we have cells of the immune system that bind to the IgE and release substances that will kill these great big worms. So I just think that is so cool. And that's why sometimes, you guys, if you have a patient with a helminth, a parasitic worm infection, you'll sometimes see an increase in eosinophils when you do a complete blood cell count. Like an eosinophilia, you're like, oh, do we have some kind of worm or parasites going on here? Great. But folks, there's a downside of this. And those of you that maybe had a chance to read that paper on how, like in some so-called developing countries, we become almost like too hygienic, right? We aren't getting our kids exposed to enough good microbes and everything. And one thing, you guys, thank heavens, because our water is safer now, our food is safer, not with fecal contamination. We probably don't have the load of parasitic worms that maybe several generations ago, people would have had more parasitic worms, right? Well, you might think that's good. I mean, I think that's good. But there's a hypothesis that the immune system, I know this is silly, you guys, but I love to think of the immune system as having a personality, that because this branch of the immune system isn't kept busy enough, it gets into mischief. You do not want a bored immune system. And the reason we say that this arm of the immune system now is causing problems because the IgE is also associated with serious, potentially life-threatening type 1 hypersensitivity reactions. These are inappropriate immune responses that can actually kill us. And the story behind this, you guys, is fascinating. So let me get rid of my worm, because probably in the United States, we associate IgE more with these allergic reactions. And ANP, you guys, I think they call it a type 1 hypersensitivity reaction. And I'm guessing probably almost anybody that lives in Sacramento, you probably have some allergy to some kind of plant pollen. We're like the plant pollen capital of the world, right? So let's see if we can't develop this, you guys. So in addition to eosinophils having Fc receptors. There's other cells of our immune system called basophils or mast cells. These are two different cells, you guys, but our basophils and mast cells are chock full of these little vesicles that are chock full of inflammatory mediators like histamine. So normally it's not a problem as long as these little cells are keeping the inflammatory mediators inside the cell, right? As long as they're not releasing them, it's not a big deal. But the challenge here, you guys, is basophils and mast cells, they have Fc receptors for which antibody. Yeah, there you go. So for some reason, and I don't get it, you guys, it must have something to do with genetics. When we are sometimes exposed to some antigen, like an antigen like a plant pollen, right? Because plant pollen, we don't make it. It's considered foreign to our immune system. For some reason, we'll end up making IgE against the plant pollen. And when we first make the IgE, it's not a big deal. This is what we call being sensitized. Because during the sensitization process, we make IgE against the plant pollen. And we'll use plant pollen, you guys, just like a little triangle. Okay, so now we've got IgE that can bind to the plant pollen. And again, folks, what happens, the IgE binds to the mast cells backwards, right? So they're binding by their little Fc stems. So then now you're sensitized. You don't know it yet, but you're sensitized. So then the second exposure to the plant pollen, what happens? Where will the plant pollen bind? It's going to bind to your IgE, right? Does that make sense, you guys? And what happens is when the IgE binds a plant pollen, there's a cascade of signals that causes these vesicles to fuse with the cell membrane and release what? Yeah, all those inflammatory mediators. So you get this massive histamine inflammatory mediators release, right? So you guys, so an antigen then that binds the IgE and triggers release of these inflammatory mediators from basophils or mast cells, we have a fancy name for it. It's called an allergen. Right? And it's triggering an allergic reaction, all these inflammatory mediators. Now, you guys, it might not be that serious because for most of us probably have a little bit of like what people sometimes call hay fever, and that's an allergic reaction in the upper respiratory tract, often just like in the mucous membranes of your nose. So you get release of inflammatory mediators, and you go through all the processes, the vascular changes, the blood vessels dilate, they get leaky, so you have swelling. That's why you feel all stuffed up, like you can't breathe through your nose, right? You might have itchy, watery eyes. But you guys, just up in your nose, it's uncomfortable, but it's not going to kill you. But, folks, the problem is that this might not be localized, or it might be in an area where the changes in the blood vessels can cause really serious consequences. Like, for example, you guys, in asthma, what's happening is we inhale the allergen and it's getting down in the lower respiratory tract and causing this massive release of inflammatory mediators. And then what can happen is we can get constriction of the airways, right? So you can't get as much air down there. And then all the changes in response to the inflammatory mediators, you get swelling and even fluid can get into the lungs, right? So you guys, can asthma be life-threatening? Yeah, right? And the thing is, you guys, what we're seeing again, it seems like more and more of our kids are developing asthma. Like in our family, nobody has asthma, but both of our kids have asthma, right? So, I mean, what the heck, what's going on there, right? So again, they're wondering if this might be a reflection of kind of a bored immune system, right? So, on an aside, you guys, and I should have brought this up sooner so you guys could look into it. So we would say these hypersensitivity, these allergic reactions, and some of the autoimmune reactions, you guys, we would say this is kind of the dark side of the immune system, right? Where the immune system is actually harming us, right? And so what some people are experimenting with, and you guys do not buy things over the internet, touch base with a medical center, but what some people are experimenting with is seeing what if we reintroduce like those helminth antigens, right? So don't buy them on the internet, you guys. People are selling worms on the internet. Don't buy those, right? If you're interested in this process, what they're exploring is by introducing the parasitic worm antigens back into a person, can you control some of these inappropriate immune responses, some of the autoimmune responses, some of the hypersensitivity reactions? And you guys, I think it's fascinating, but you want to make sure that it's not you're doing it yourself in your garage kind of experiments. Make sure you connect like with a good medical center that's carrying out really responsible research on this. I think it's really fascinating. I think it might offer some help in the future to control some of these immune disorders that we seem to be seeing more and more of. Yeah. Okay. All right. So, any questions, you guys, on IgE? Okay. And then, folks, IgG, don't even worry about it. They find it on the surface of B lymphocytes, so some people think it might be a B lymphocyte receptor. But you guys, I won't even ask it on the final exam, so we'll just leave it at that. There's enough other stuff here to remember. Okay, so you guys, let's see, we've got about like 20 minutes left, something like that. We can do this. All right, folks, so if I ask you on the final, which cells make antibodies, what are you going to tell me? B lymphocytes, yeah. And folks, don't worry about this, but you might read about it. So when a B lymphocyte starts making antibody, we call it a plasma cell. Excuse me. So you guys, a plasma cell is a B lymphocyte that's actually making antibodies. Okay. And what we next want to do is take a look, you guys, and see, we have all hundreds, thousands of different lymphocytes circulating in our blood or in our lymph nodes and spleen. And these B lymphocytes, they have surface receptors to bind to very specific antigenic determinants or epitopes. And so what we want to explore, you guys, is how does a B lymphocyte know to start making antibody, right? So this, so first of all, you guys, B lymphocytes, like all blood cells, are made in the bone marrow. Okay, so that's where all of our blood cells arise, is in the bone marrow, so that's where they're coming. And again, you guys, they're different B cells will have different surface receptors to bind to different antigenic determinants. So I'm going to make this pretty simple, you guys. Let's just say we have two different B cells. So we'll call this B1, and it has a surface B cell receptor. And they are often antibodies, you guys, so either IgM or like I said, IgD. So let's say these are the B cell receptors for B1, and then folks, I'll keep it really simple. Then I'll make another lymphocyte B2, and I'll have a little bit different B cell receptor here. Okay. All right, and they are not active right now, right? They're just resting. They're waiting for some signals. And you guys, we're going to keep this really simple. We're going to say before they can start making antibodies, they have to receive two signals. And we're going to say the two signals is, the first signal is they have to bind to antigen, or the complementary antigenic determinant. And then you guys, we're going to say the second signal will be T helper cytokines. Okay, so we're keeping this really simple. So these guys are just hanging out. They're not doing much. But then, folks, let's say that we get invaded by some pathogen, maybe some bacterial pathogen, and maybe the bacterial pathogen makes a protein, right? And maybe as the pathogens are growing and dividing, and maybe some of them are dying, they release the protein, right? So here's the microbe protein. Right? And this will be the antigen. It's going to trigger the immune response. Now, the way it's going to do that is it's going to bind to the B lymphocyte that has the complementary surface receptor. So you guys, so if this is our antigen right here, these little triangles, which B lymphocyte will the antigen bind to, B1 or B2? B1, and you guys just knew that instinctively, right? So you're right, you guys. The antigen chooses or select. The antigen chooses or selects the specific B lymphocyte to activate. Right? So that's the first signal, right? The first signal is the binding of the antigen. And then you guys remember how I said we're going to really simplify this. The second signal is going to be what? Yeah, if in doubt, say T helper cytokines. Okay, and again, you guys, we're not cartooning it all. I think in A&P they go into a lot more delicious detail than this, so this is really super fun. Okay, so you guys, so with these two signals then, with antigen binding and T helper cytokines, it's going to trigger this B cell to start proliferating. It's going to start dividing like crazy. So the B cell will start division, giving rise to a platoon of little genetic clones. And so you guys, I'm going to make my little B1 little genetic clones here. And notice, you guys, I have a bunch here, and I'm going to put a couple over here, because it turns out there's two subpopulations that have different functions. So I'm going to put a little platoon over here, right? But these are all little clones of that original B lymphocyte that got activated. So you guys, this group over here, these are going to be our plasma cells. And what is a plasma cell? That is right. That is right, you guys. So our plasma cells here are going to start pumping out what? Yeah. And you guys, at first, they're going to make IgM. But remember, you guys, we got our T helper there. T helpers trigger class switching. So what will they now make? IgG, the best antibody, right, you guys? So I'm going to show them just pumping out little IgG. And you're like, well, how does that help? Oh, this is so elegant. Your immune system is amazing. So you guys, this IgE is going to bind to what? Yeah, the invading pathogens, right? The pathogen that triggered the antibody production, that's a pathogen that's going to be attacked by those antibodies. Isn't that? It's so elegant. It's so beautiful. So you guys, so tell me, why is that going to be helpful? The antibodies binding to the pathogen. How is that helpful? Oh, you nailed it. Neutralization, agglutination, opsonization, complement activation. And what if this was a pregnant mom? Transplacental transfer, right? So that baby's going to have those protective antibodies on board. So isn't that just, oh my gosh, you guys, it's like chocolate. It's so beautiful, right? So the antigen chooses which B lymphocyte will be activated. The B lymphocyte, with help of T helpers, starts dividing, forming these little clones. And again, with the T helpers, undergoes class switching IgM to IgG. But you guys, what about these guys who are over here? Why else are T helper cytokines important? What else do we want to be making? You nailed it, you guys. You got it. Here are our beautiful B memory cells. And their job is, they're not going to make antibodies. Their job is to live long and remember that antigen. So that if subsequently we get reinfected with that same pathogen, you guys, these B memory cells, they're pre-primed. They are ready to blast into action. So these memory cells will be responsible for what's called the secondary immune response. And we'll see, you guys, we have another slide here that will explore this secondary immune response. And the secondary immune response, you guys, the advantage of the secondary immune response compared to the primary immune response, which is the first time we encounter the pathogen or the toxin, the secondary immune response is going to be faster, stronger, longer lasting, and we're going to see a big increase in IgG production. So it's going to be more protective. So that's the beauty of those memory cells. Again, they're pre-primed, so if we ever encounter the same pathogen or toxin again, we get a faster, stronger, longer lasting, more effective, primarily IgG response. And you guys, that's the basis of Vaccination. What do we want in vaccination? What do we want to make? Yeah, that's a goal, right? And also, you guys, it's the basis of lifelong immunity. Like if in the old days, say that as little kids, you guys, we got infected, say with smallpox, and we survived, right? We would never get infected with smallpox again, and why? Because we made all these all these memory cells. Yeah. So you guys, that's kind of the overview here. This, of course, is a nicer cartoon. So you guys, here are the resting B lymphocytes with their B cell receptors. Here's the antigen binding to selecting the correct B lymphocyte. They start proliferating. We end up with two subpopulations of plasma cells pumping out antibodies, and then we have our memory cells. And again, you guys, these memory cells, they'll live for years, maybe decades. So again, if you ever encounter that same pathogen, the memory cells are pre-primed, right? They start replicating faster. We're going to have more plasma cells. We're going to have more memory cells, and we'll have faster class switching to IgG. And we'll see, you guys, those memory cells will trigger higher antibody production. Let me just switch to the slide. Even though graphs are boring, you guys, this one's kind of nice. So you guys, here we're exploring the primary and secondary immune response and humoral immunity. We're looking at antibody production as our response. And I like this, you guys, because here we're looking at the antibodies produced in response to a tetanus vaccine. So we're injecting our patient with tetanus toxoid. And what we can see here, you guys, there's this long lag period, because that takes a lot of time, right? That's going to take like 10 to 14 days for all those cells to communicate with one another. So the first time you encounter an antigen, there's this long lag period. It's slow, right? And then what we'll see is a little peak of IgM, and remember, that's not the best one. And then later, like 2 weeks later, we finally get a peak of IgG, our best antibody. You guys notice it's not very high antibody production. It doesn't last very long, right? And it's slow. But you guys, what is so cool? What did that tetanus toxoid, what did it trigger production of? These guys right there. That's what we're after, right? So if subsequently the vaccinated person steps on that proverbial rusty nail, right? Now those memory cells are pre-primed, and they start dividing, they become activated right off the bat. So what we'll see, you guys, is a faster response, right? A higher antibody response, so faster, stronger, more antibodies made, primarily what? IgG, it'll last longer, right? And so this is going to be the secondary response, faster, stronger, longer lasting, primarily IgG, that will protect you from serious harm, from either an invading pathogen or, say, from an exotoxin like tetanus toxin. Yeah. So the secondary immune response, you get the basis of vaccination and lifelong immunity. Who's responsible for the secondary immune response? Those beautiful memory cells. Yeah. And you guys, can we make memory cells without T helpers? No. So you guys, if you have a patient, say I'm now experiencing AIDS, would vaccinating me make a lot of sense? Well, you could vaccinate me, but I'm not going to make any memory cells, right? So really tough. Oh my gosh. All right. And then, folks, this will be these last few slides, you guys. The details we haven't discussed will be bonus. Right? And like yesterday, the poor folks, they were just so tired. And this is a cool topic, you guys, but it's a lot to think about. So this discussion of T-dependent and T-independent antigens will be totally bonus on the exam, right? But it's kind of like a good news story. So that's why I just wanted to share it with you really quick. So what we've been describing, folks, so far are what we call T-dependent antigens. Antigens that recruit T helpers to get involved. Antigens that trigger T helpers to release those cytokines, right? Those are called T-dependent antigens. They are the best antigens, and they're usually proteins. But, folks, there's problems. There are problems because what about an encapsulated bacterial pathogen? Okay, so here's the bacterium, and it's got that thick capsule, right? And the capsules usually are made out of polysaccharides. So you guys, some examples we've mentioned before of these encapsulated pathogens include Neisseria meningitidis, that causes bacterial meningitis in young people like you, Haemophilus influenza, which can cause middle ear infections and meningitis in babies, and then Streptococcus pneumonia, which is a common cause of bacterial pneumonia following, say, influenza, and can also invade the central nervous system. These stinkers all have a capsular polysaccharide. And what do we know about that polysaccharide, you guys? What do our phagocytes do with those encapsulated bacteria? Nothing, right? Because our phagocytes don't have receptors for capsular polysaccharides. They can't phagocytize them. So you'd say, well, shoot, let's make a vaccine. Let's get a vaccine that will trigger IgG production, right? The problem is, you guys, polysaccharides won't trigger IgG production because they don't recruit T helpers. Okay? So let me see if I can explain this to you guys. So this capsular polysaccharide is an example of what's called a T-independent antigen, meaning they don't recruit T helpers. T helper cytokines aren't going to be produced. And these are bad antigens. Not good, not good. And the reason is, what they do is say this is a B cell, a B lymphocyte, right? And let's say the B lymphocyte has surface receptors to bind to monosaccharides, okay? And this will be the cartoon, you guys, of our capsular polysaccharide. They're usually chains of the same sugar over and over and over again. So the bad news is these capsular polysaccharides, what they do is they can bind to a single B lymphocyte and cross-link receptors. So this is the capsular polysaccharide binding to and cross-linking these B cell receptors. And what that can do is it can trigger the B lymphocyte. It will start making IgM. Briefly, for a short period of time. But you guys, there's no T helpers involved. So can this B lymphocyte undergo class switching from IgM to IgG? No, there's no class switching. And you guys, remember, you guys, IgM, it's not opsonic, right? And we need some opsonic antibodies here, right? But darn, what are we going to do? No T helpers, no IgG. And what about, if we're vaccinating, what do we want? No memory cells, right? So that's horrible. And to make it worse, like the very old and the very young, we just don't respond to these T-independent. I mean, there's like nothing, right? So this is really serious. How do you develop a vaccine against these really serious encapsulated bacteria? And you guys, this is where it was brilliant. So what did the folks do, the smart folks? They're like, well, let's take the capsular polysaccharide and link it to a protein. And this combination, you guys, the capsular polysaccharide linked to the protein, it's called a conjugate vaccine. So this whole thing is called a conjugate vaccine. And the beauty, you guys, of conjugate vaccines, if you now inject your patient with them, the polysaccharide gets processed as if it was a T-dependent antigen, right? And that's gorgeous, right? So now what do we get? We get IgM to IgG class switching. And remember, you guys, there's our opsonic antibodies. We're going to make memory cells. And very importantly, you guys, the young and the old can respond. So and this is what I wanted to kind of, almost at the end, finish with this. So you guys, in previous generations, a lot of people died. Young people died from bacterial meningitis. A lot of babies died from Haemophilus influenza. A lot of old people and young people died from strep pneumonia. But now, folks, we've got conjugate vaccines. You guys can get vaccinated with a conjugate vaccine against Neisseria meningitidis. The HIB vaccine, you guys, those of you that have babies, that's a conjugate vaccine. There's conjugate vaccines to protect us old folks from strep pneumonia. So that's just such good news, folks. And I know you guys, your bottoms are sore and you're exhausted, but let me just hit 2 last bonus topics really quick, okay? And Professor Johnson said she wouldn't yell at me if I run over. So guys, so bonus topics, okay, the extra information we give here, bonus topics. So finish humoral immunity. Now we want to go just so briefly over cell-mediated immunity. You guys, the branch of the immune system to protect us against intracellular pathogens. So we'll look at activation of macrophages by T helpers, and then we're going to look at cytotoxic T lymphocytes killing virus-infected cells. And this is going to be so fast, you guys. But again, remember, it's bonus. So you guys, with we actually covered most of this. So you guys, with activation of the resting macrophages, remember they can get parasitized by like Mycobacterium tuberculosis, and while they're still resting, they can't kill them. So the T helpers can produce those cytokines like gamma interferon that will activate the macrophage, turn them into angry killers, right? And with those angry killing macrophages, they're going to phagocytize more quickly. They're going to increase production of those hydrolytic digestive enzymes of the lysosome, and they're going to increase production of those toxic reactive oxygen intermediates like the superoxide radicals, the free radicals, hydrogen peroxide, right? So if we can activate the macrophages quickly enough, they can kill those facultative intracellular parasites. Yeah, but again, did we need T helpers? We sure did, yeah. And the last example, folks, here is of the cytotoxic T lymphocytes. And again, they require T helper cytokines to carry out their job of killing our cells that have been infected with pathogens like viruses, yeah? And you guys, what's so cool is with the help of T helper cytokines, and again, you guys, we'll do this very superficially. Let's say this is our cell that's been infected by a virus. Our cells will present some of the viral antigens on the surface of the cell to mark that cell for destruction, right? And here's our beautiful cytotoxic T lymphocyte. It will bind to the viral antigen that's being presented on the surface of our cell. And then in response, you guys, binding the antigen and the T helper cytokines, it releases what they call the kiss of death. And the kiss of death, you guys, includes perforin. This is a lot like the membrane attack complex of complement. So perforin, you guys, would insert into the cell membrane of the virus-infected cell. You might lyse it right off the bat. But if that doesn't work, then another component of the kiss of death is called granzyme. And what granzyme can do is it can get into the inside of the virus-infected cell through those perforin pores, and the granzyme triggers programmed cell deaths, sometimes called cell suicide or apoptosis. It will trigger the cell to kill itself. Now, is that going to cause tissue damage? Yes, it is. But what's the advantage, you guys? We can kill this cell early enough, we can stop the viruses from replicating and spreading. Yeah? Does this require T helper cytokines? Yes, it does. Okay, so you guys, that will be a bonus question. Okay, so again, I know you're overwhelmed, so try not to stress about it. Just a bonus question. And you guys, please keep checking Canvas just for any updates on the final exam. Okay, and you guys try to eat, try to sleep before the final exam. Remember, it starts at 10:15 next Tuesday. And folks, I do have your lecture exam 3 up here. I'll bring them down to lab if you want to wait, and I'll pass them out in lab. Okay? You guys take good care.